Good morning. So first of all, I want to thank Chris and Neuronet for inviting me here and facilitating my venue to this very nice conference. The consortium I will present is just starting, so I'm not going to present much data, but mainly objectives and people. So the title, do you hear me well? The title of the consortium is IM2PACT, for investigating mechanisms and models predictive of accessibility of therapeutics into the brain. So the rationale for starting this project is the following. The blood-brain barrier remains a major obstacle for biologics, and today it's actually thought that it's one of the main reasons why there are so few biologics on the market presently. There are a few reports in the literature, a lot of them actually, about varying biotherapeutics across the blood-brain barrier, but most of them are using the transferrin receptor as a carrier, and there is clearly a need for more brain-specific carriers. Another aspect of the rationale was the fact that today there is no predictive blood-brain barrier model, in particular for predicting in vivo exposure of biologics. There is a lot of, and there is more and more evidence also reporting that blood-brain barrier dysfunction is probably at the origin, at the etiology of a lot of neurological diseases. And these are actually also part of this vascular hypothesis of pathogenesis of neurovascular diseases. And so the idea was to bring a pre-competitive consortium to further the understanding of the blood-brain barrier in health and disease, and for the identification of innovative brain delivery systems. And we wanted, in parallel, to synergize and capitalize on findings from other relevant IMI initiatives such as IM2PACT and IBISC. So the objectives, the principal objectives of this consortium is actually to discover innovative and efficacious brain delivery systems, to characterize best state-of-the-art blood-brain barrier models with good predictivity, identification of translational readouts that are closer to the pathogenesis of neurodegeneration and mimicking altered BBB under disease condition, and finally, an in-depth understanding of the biology of the blood-brain barrier and transport mechanisms. So I am going to go through the 5 work packages, actually 6 work packages of this project. I have indented pictures of the leaders, of course, and I will show a little bit later on who are the partners, the whole partners of the project. So the idea of the work package 1 is to identify new targets and mechanisms for blood-brain barrier function in disease and in healthy brain tissue. And so the idea here is to, there are 2 main packages actually. We are starting with analyzing existing databases and integrating these to actually identify targets that have not been looked at previously. And in parallel, we are doing experiments on brain microvasculature based from primary cells from human tissues, and I'm not going to the detail of these sketches, but the idea is to do proteomic studies and transcriptomic studies on these tissues to identify candidates that are disease associated or differentially expressed in either disease versus health or in brain versus peripheral tissues. So this is work package 1. Work package 2 will deal with establishing iPSC-based endothelial cells for phenotypic validation. So the idea here is to generate endothelial cells from iPSCs, and this is not as easy as it seems from looking at the literature. And then from what will be learned from work package 1 to induce the mutations of the genes and pathways that have been discovered in work package 1 to evidence a phenotype after these mutations. And so the idea here is to validate disease-specific or differential expressed genes or pathways of potential relevance to brain transport or to brain to neurodegeneration. Work package 3 is dealing with establishing best state-of-the-art human iPSC-based blood-brain barrier models. And so this is intimately working with work package 2 into producing differentiated brain endothelial cells from human iPSCs, setting up mono or co-cultures or 3D settings or microfluidics. We have actually 5 models that we are funneling down to select the best one. In parallel, we are planning to do some mathematical modeling of receptor or carrier-mediated transcytosis across the blood-brain barrier. So here, as I said, the delivery is the best state-of-the-art model to really be able to predict, because today there is no model that is able to predict the exposure of biotherapeutic. Work package number 4 is a little bit apart from the other ones, but also makes a lot of sense. The idea here is to explore neurotrophic viruses and neurotrophic AAVs to try to understand their mechanism of brain penetration and build on this mechanism some potential delivery systems. So we are looking at neurotrophic viruses such as flaviviruses and AAVs such as AAV9 or others. And finally, the work package 5 is the one that is going to validate the targets that will have been shown in the previous packages. For this validation, there will be a need for each blood-brain barrier target. There will be a need to build tools, either viral particles, antibodies, peptides, in vitro models, mutated for the target, small molecule. It will, of course, depend on what target will be identified. Even maybe probably models, transgenic models of the target. And so there are the final delivery of the proposal, which are either a validated target for brain delivery or a validated target for curing, for addressing neurovascular or metabolic diseases. Because I forgot to say, but I will come back to it, which diseases we are especially focusing on. I don't want to leave out one of the very important work package, which is not doing science, but has a lot of importance in our group, of course, which is the management, the communication, and dissemination. And Philippe is really a great person to do this. I don't think that it's necessary to go through this, but this is very well articulated, at least on paper, and we will see how it will work in reality. But so every work packages can start at the same time with different tasks, and little by little they will exchange the iPSC cells. Once they are ready, they will be passed to work package 3 to apply to the blood-brain barrier models. The targets, the genes that are selected will be passed to work package 2 to induce the mutations. They will also be passed to work package 5 to generate the tools and models for validation, and so on and so on. I think that we will skip the rest. So now a quick overview of the different partners that are involved into this consortium and what they bring into this consortium. So we have a very nice set of academic groups which bring to the consortium their know-how on blood-brain barrier and disease models, their characterization of mechanisms of brain transport, of virus-mediated transport, their expertise in establishing iPSC-based models, and several of them are very experienced already with models of the neurovascular unit and the blood-brain barrier model. We also have, and that's a great asset for a consortium, several biotechs, which are actually bringing to the pot their know-how in iPSCs or progenitor-derived cells, innovative technologies, new tools, And finally, we have a certain, a group of large pharma companies which are going to be most active into understanding the preclinical and clinical consequences of the disease-modified blood-brain barrier, analytical and chemical resources, powerful and connected bioinformatics, and of course a direct link into the clinic. I don't want to forget to mention that we also have very nice experts, Danica Stanimirovic, Jean-Michel Sherman, Christine Mumry, David Begley, and Niels Hopper are all very specialized experts in the field of blood-brain barrier and iPSCs. The diseases that we are considering, because we had to limit ourselves, are listed here. So we are looking at neurodegenerative diseases, ADPD, ALS, and MS, and metabolic diseases with an impact on CNS, such as diabetes and obesity. I guess the rest I had more or less said. I mean, this is all theoretical things that the industry companies are able to bring to this consortium. And what we expect as an impact for the project is better translational tools and models to assess efficacy, biomarkers for disease, clearly linked to clinical relevance, and better models, including in silico models for predicting blood-brain barrier permeability. And on disease, we are hoping to have a few new delivery systems and/or therapies for diseases of this topic text. And having said that, I want to thank you all for your attention, and if there are any questions that I can answer, I'll be glad to do so.